to today's webinar. I'm Kelly McSweeney, contributing editor at CNEN Media, and I will be moderating today's event. This webinar is titled Screening Methods to Identify RNA Targeting Small Molecules and is being sponsored by WuXi AppTec. CNEN works with sponsors to identify topics that are of interest and value to CNEN's audience and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the top right corner of the screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You are encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. Please note that CNEN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at CNN Online after the live webcast. The presentation today is being sponsored by WuXi AppTec. WuXi AppTec is a global company that provides a broad portfolio of R&D and manufacturing services that enable companies in the pharmaceutical, biotech, and medical device industries to advance discoveries and deliver groundbreaking treatments to patients. During the presentation, we will hear from Dr. Zhifeng Yu. I will now hand it over to Dr. Yu.
Today's presentation aims to introduce our expertise in RNA targeting small molecule discovery. By the end of this webinar, we trust you will have a clear understanding of how we can enhance your RNA drug discovery efforts. Drug discovery has a long history, and it can be dated back to the early days of human civilization. From early drugs derived from plants, animals, and minerals, to the recent synthetic drugs developed using modern chemical methods. As a result of advances in technologies in chemistry, biology, and etc., the early random approach to drug discovery was replaced by a more rational approach that involves the element of design. WuXi AppTec is a CRO company dedicated to contributing to the global healthcare community. Our department is focused on speeding up the process of searching for novel therapeutics by developing new technologies and platforms, including targeting RNAs. So, can RNA be a drug target? Well, we know that over 90% of drugs approved by FDA modulate the actions of proteins. But from this figure, you can see that only 1.5% of the human genome encodes proteins, where only a very small fraction is druggable. So, the community is always exploring novel therapeutic targets. And from the left side, landmark events and key milestones from historical RNA-DNA research indicate the development of oligonucleotide therapeutics to target RNAs. In principle, oligonucleotides can be rationally designed against virtually any genetic target. However, these nucleotide therapies involve large and highly charged molecules which are difficult to be delivered. So given the choice between an oligonucleotide and a hypothetical small molecule, small molecules would be preferred in many cases. But still, the question is, can RNA be targeted by small molecules? More and more recent studies actually indicate that messenger and non-coding RNAs contain highly structured elements, and many of these structures are important for their functions. Therefore, these RNAs can be potentially targeted by small molecules. because they have a binding pocket. And indeed, since 2017, nearly $1 billion have been invested to discover small molecules that can target RNAs. You can see from this slide, investors and big pharma companies have invested quite a lot of money to finance emerging companies to enable RNA technologies and focus on RNA targeting small molecules. However, we have to say RNA targeting is challenging, mainly because of the less investigated molecular biology, biochemistry, structural biology, and pharmacology for potential RNA targets. But I think the most frequent concern people have when they start targeting RNA is whether RNA is less druggable than proteins. Current evidence does suggest that bacterial, viral, and mammalian RNAs fold back on themselves and form complex structures. In this review paper by Dr. Kevin Weeks, an algorithm called POC Finder was applied to estimate the quality of the potential ligand binding pockets of a set of RNAs. Similar to protein targets, different RNAs present different qualities of pockets in terms of small molecule binding. Interestingly, molecules targeting higher order tertiary RNA structures are more drug-like, while compounds targeting RNA secondary structures or repeated sequences are difficult to achieve good drug likeness and high potency simultaneously. To date, quite several RNA targeting small molecules have been identified that use a range of action mechanisms, including directing splicing, for example, by acting as molecular glues with cellular proteins, or inhibition of the translation of undruggable proteins, and deactivation of functional structures in non-coding RNAs. These molecules have been demonstrated to regulate diverse biological processes, with implication in treating different human diseases, such as riboswitch and other synthetic ligands as riboswitch inhibitors in inhibiting bacterial translation in infectious diseases, or risdiplam as SMN2 binder in facilitating SMN2 alternative pre-mRNA splicing and restoring SMN protein expression in spinal muscular atrophy. Our department, WuXi Biology, with the scope of providing a full spectrum of biological services and solutions to support standalone and integrated projects, has developed capabilities, products, and services to facilitate our clients in their RNA targeting research. We strive to offer a comprehensive solution for your specific target, covering everything from target verification, hit identification, to lead confirmation. This slide is our general workflow for RNA drug discovery, designed to offer a streamlined solution for your specific RNA target of interest. We initiate the process by consulting relevant literature for the specified client RNA target, followed by commencing RNA synthesis and characterization. We investigate the folding of your RNA. We test their stability and integrity. We are familiar with using the ShapeMap technology to verify RNA secondary structure to predict the quality. And once both parties are satisfied with this RNA quality, we will run affinity-based drug screening to identify small molecule binders. These potential small molecule hits can be resynthesized and assay validated at WuXi AppTec. We have a bunch of biophysical assays to directly confirm the binding. If your RNA is well studied, we can develop potential biochemical and cellular assays to further confirm the biological function of those small molecules. We use screening historical data and RNA-seq to evaluate the off-target effect and try our best to provide ligands with specificity to RNA targets. Here is our toolbox specialized for screening RNA targets. Our RNA drug discovery is derived from our small molecule library collections, including DNA-encoded libraries, fragment libraries, and traditional small molecule HTS libraries. With zipper DNA-encoded library selection by physical binding assays and ASMS using the high-resolution mass spec, by using those methods, these libraries have been applied to screen RNA targets. And by using this robust screening process, particularly our DAO selection, extensive datasets rich in binding information have been generated. To leverage this, we have established an integrated machine learning pipeline based on our screening data. This allows our trained models to validate screenings and pinpoint novel binders. Our validation process for small molecule RNA binders involves orthogonal assays. Additionally, we are equipped to conduct a range of biophysical binding assays efficiently. And for swift structure and binding site verification, we do ShapeMap. ShapeMap is an innovative technology that employs shaped chemical probes to selectively and covalently label RNA functional motifs. These probes induce mutations during reverse transcription, which can then be identified through sequencing, providing valuable insights into RNA secondary structures. This service has gained widespread popularity within our RNA platform. It offers a rapid assessment of your RNA target's folding and aids in finding the potential ligand binding sites. ShapeMap is now a very important approach to assess the quality of the RNA preparation for the screening. Our capabilities extend to both in vitro and in-cell probing. You can expect to receive your data within a turnaround time of approximately 2 to 4 weeks. Regarding the several screening approaches against RNA, I will focus today mainly on DAO. So what's DAO? DAO is DNA-encoded libraries, a technology where genetics and small molecules are linked together to allow the synthesis and screening of billions of molecules through a biophysical affinity assay. Since the publication of the first DAO paper by Dr. Richard Lerner, DAO was introduced to pharmaceutical industries and later entered a stage of rapid development, particularly when Illumina launched the next-generation sequencing, which enabled a dramatic rise of data output and falling cost of sequencing in 2005. DAO is now widely used as an early drug discovery platform in academia and industry, and more hits were discovered by using this technology. Our company started working on DAO in 2017, and within these 6 years, we have become one of the major service and product providers in this field. The typical DAO approach encompasses the synthesis and application of DAO libraries. By employing combinatorial chemistry and employing split and pool strategies, chemists can efficiently generate millions of unique DAO molecules within a span of several months. Each DAL molecule is equipped with a distinct DNA fragment that's covalently linked, serving as a molecular barcode. These molecular barcodes empower the pooling of billions of diverse DAL molecules into a single Eppendorf tube, from which they can be selected by a recombinant target. The DAL selection process is straightforward. It needs the target to possess an affinity tag, allowing it to be immobilized on the affinity matrix. We use magnetic beads. This, in turn, facilitates the separation of non-binders from binders through bead washing. Following this, we subject the target to heat denaturation, releasing binders for quantification, and this cycle can be repeated for 2 to 3 rounds to increase the difference between binders and non-binders. And subsequently, we conduct PCR and employ next-generation sequencing to identify the DNA barcode sequences and align these sequences with their respective chemical structures. This method yields a wealth of SAR information, enabling the identification of compounds for off-DNA resynthesis and subsequent validation. So, with this technology, we have synthesized several hundred billion DAL molecules, classified them into distinct business models tailored to meet our clients' diverse needs. This slide offers an overview of our DAL modalities designed for different screening purposes. We have the classic traditional DAL, presenting an impressive collection of up to 90 billion unique compounds. These libraries have been applied in quite a lot of DAL campaigns screening for novel ligand against several hundred client targets, primarily proteins, and with a few targeting RNAs. Additionally, we offer covalent DAL featuring molecules equipped with either irreversible or reversible warheads. Currently, our covalent DAL molecules are optimized for targeting cysteine residues only. However, we are actively working on expanding our covalent DAL libraries to target other amino acids in the future. We have devised specialized screening methods for these covalent DAL libraries to identify irreversible binders effectively. Another noteworthy offering is our microcycle peptide-based DAL, incorporating a percentage of library members with peptoid-like structures. Within these microcycle DAL libraries, we have included linear peptide DAL as controls for comparative analysis. Notably, we recently published a paper showcasing potent compounds identified from these peptide DAL libraries, demonstrating their efficacy in inhibiting MDM2 p53 interaction. Well, the unique architecture of DAL makes this technology a great platform to screen for PROTACs or other tags for degradation. Our bifunctional DAL libraries leverage the flexibility of E3 ligands in tandem with various combinations of DAL molecules and linkers to pinpoint small molecules that facilitate the degradation of proteins and RNAs. We employ very flexible business models to provide DAL screening services. DAL Pro is our full service, where DAL libraries with the highest diversity and best novelty will be offered. We need to request target information for this service so as to extensively coordinate with clients and design a screening strategy accordingly. All the experiments of a DAL Pro campaign are performed by experienced WuXi DAL selection scientists. And in the end, 20 off-DNA resynthesized compounds identified from DAL Pro will be delivered for clients' further validation. However, if the client, for some reason, cannot disclose their target information, we would alternatively provide the DAL Lite service, in which DAL molecules and screening methods will be wrapped as a kit and sent to the client. Clients can follow our SOP to run DAL selection in their own laboratory. They will need to send the post-selection DAL samples back to our facility, and these samples will be further processed, sequenced, and analyzed. A data report containing the structural information of all positive binders will finally be delivered. We have global sites to support clients from different regions, and in each site, a teaching lab is well equipped to accommodate clients where they can learn how to run DAL selections. As a screening platform, we consistently deliver diverse and high-quality chemical space. For DAL, we are making improvements every year to enhance their drug likeness and diversity. And other than DAL, the other two libraries we can offer to screen RNAs are Fragment libraries and HTS libraries. And from the right side of this slide, you can see our 3000 fragment library. Each fragment in this library has been verified by LC-mass to guarantee their quality. And all these fragments follow the components of the rule of three, so the quality of this fragment library is guaranteed. I'm a DAL selection scientist. However, I must acknowledge that HTS screening is still a pivotal platform in early drug discovery. Our department has a 300,000 small molecule HTS library collection, either synthesized by WuXi or purchased from other vendors. These compounds were well selected to ensure that pan-assay interference compounds and biologically useless compounds were not included. Our libraries offer guaranteed diversity to significantly increase the chances for our clients to discover binders for their various target classes. And we have developed ALICE ASMS to search these compounds against RNA targets. I want to demonstrate our capabilities in finding RNA-binding small molecules by using these libraries. Today's focus is DAL, but still other methods like ALICE ASMS have been employed for compound validation. SMN2 messenger RNA was chosen for this POC. It is well known that SMA is caused by deletions or homozygous mutations in the SMN1 gene. Another copy of SMN1 is SMN2. These two genes are nearly identical. The only difference in their coding region is a translationally silent C to T transition in SMN2's exon 7, which results in different alternative splicing, producing an inactive C-terminally truncated SMN protein. Therefore, promoting SMN2 exon 7 inclusion can compensate for the lack of SMN1. Several compounds have been identified as effective SMN2 exon 7 splicing regulators, mainly through splicing reporter phenotypic screenings. The famous compound risdiplam has been approved by FDA to treat SMA and binds directly to SMN2 exon 7. Therefore, target-based approaches are promising to identify small molecules that can directly bind to SMN2 messenger RNA. More importantly, biochemical and cellular assays have been reported to test the efficacy of these compounds in vitro. So, we synthesized an SMN2 mRNA fragment spanning exon 7 and adjacent introns. Following conditions published in literature, this RNA was folded in a slowly cooling down process. To verify the folding, we performed shape map to evaluate SMN2 secondary structure. Our data identified several stem loops that are consistent with published structures, particularly this terminal stem loop 2 located at the exon 7 and intron 7 junction with the implication of regulating SMN2 splicing. We then initiated the DAO selection to target SMN2. However, our initial attempt was not successful. What we found was that DNA barcodes of DAO molecules bound to RNA fragments based on the significant enrichment of DNA-only libraries which were spiked into our DAO superpool. We further figured out that the binding was mainly mapped to the single-stranded DNA region of the DNA barcode. This slide presents the architecture of the DNA barcode of a DAO molecule. You can see that DNA barcode is connected to the small molecules through a linker, a long flexible PEG linker. But the DNA barcode has two regions. The double-strand DNA region spanning the 5' forward priming, coding, and library ID, and the 3' single-strand DNA region spanning pool ID, degenerate sequence, and the reverse priming. The reason for this design is to facilitate primer annealing during PCR amplification, because the top and bottom DNA strand are covalently linked through the linker. Given that this single-strand DNA region can cause significant DNA-RNA interaction, to enable RNA DAO selection, we decided to convert this single-strand DNA region into double-strand DNA, and we call this specialized library zipper DAO. Well, to test whether the zipper DAO is effective in blocking DNA-RNA interaction, warheadless DNA-only libraries were utilized. And here is the data showing that single-strand DNA's motif-specific binding can be mitigated by DNA zipper treatment. We used rebel switch from different species and applied different affinity metrics to investigate the background effect. These RNAs were folded and screened in parallel with warheadless single-strand DNA DAO and the warheadless zipper DNA DAO. The sequencing data was subject to DNA binding motif analysis. From this data, we concluded that the significant interaction between rebel switch and the single-strand DNA DAO can be dramatically decreased by the zipper treatment, so that in a real DAO selection, the binding pattern of chemical warheads will not be diluted or interrupted. So from this data, what you are looking at is the lines in orange and the lines in blue. The line in orange is the single-strand DNA DAO, but the line for this blue is actually zipper DAO. Okay, we reinitiate SMN2 DAO selection by using zipper DNA DAO. DAO is an affinity binding-based screening and requires minimum assay development. But we really care about the quality of the target to be presented. This quality includes purity, stability, and activity. This slide demonstrates a typical pre-selection QC for the target to be screened. Although this is for RNA, it actually can be applied to any DNA-RNA binding proteins. Our SMN2 RNA is biotinylated, so we first confirmed that this RNA can be immobilized to streptavidin magnetic beads. Figure A is the bead capture test data running through a DNA agarose gel. You can see that biotinylated SMN2 retained on beads in length 5, but non-biotinylated RNA as a control was not captured to bits in length 6. And second, we need to confirm whether this RNA is still active when immobilized. Figure B is the two-compound binding test data. We used fluorescence indicator displacement assay. You can see that the binding of the fluorescence dye can be blocked by PK4C9 in a dose-dependent manner. PK4C9 is a published small molecule that binds to SMN2 terminal stem loop 2, therefore indicating the presence of an active binding pocket in our RNA preparation based on this data. We lastly did a RNA and DNA barcode interaction test. The warheadless DNA-only zipper DEL was selected with SMN2. The final sequencing data was analyzed to find DNA binding motifs. By comparing SMN2 selections at two different concentrations to the no target control in Figure C, you can see that there's no enhanced RNA-DNA interaction when using zipper DEL. So we move forward to screen real zipper DEL libraries against SMN2. In this selection, we had 4 conditions, SMN2 at 2 different concentrations, and we had 2 controls, the no target bit-only control to find bit binders, and the mutated G4 RNA control to help to rule out non-specific RNA binders. The sequencing data was analyzed to calculate the enrichment score, and these cubic scatter plots demonstrate the enrichment of this building block 2 and building block 3 disynsome family. This is a 3-cycle DEL library. A single dot in this cubic plot represents a full DEL molecule, while the line formed by all these dots represents all the enriched compounds containing the same building block 2 and building block 3, with only varied building block 1 from this family. This is why I said previously, DEL can provide strong SAR information directly from the screening that can further facilitate the next stage heat tri-aging and medicinal chemistry optimization. So from this data, you can see that this compound is significantly enriched in SMN2, but in the no target control and the G4 mutant control, the enrichment was either not visualized or dramatically decreased, indicating the binding might be specific to SMN2. DEL selection, all the compounds were DNA barcoded, so DEL identified compounds need to be off-DNA resynthesized and validated in another assay. Given that DEL selection is affinity binding based, we normally choose another binding assay to do the validation. SPR is frequently used to validate RNA small molecule binding. But here, we employed affinity selection mass spec, ASMS. This technology does not need the target to be tagged or immobilized. And we have Orbitrap Q and Agilent high-resolution mass spec that can detect and identify multiple compounds even when there are minor differences in their masses and when they are presented in low concentrations. With these machines, our compounds can be pulled together and screened against the target at the same time. You can see ASMS workflow from the right side of this slide, where DEL identified SMN2 compounds were pulled together with each at 10 micromolar and incubated with folded RNA. After incubation, binders were separated from non-binders by using size exclusion chromatography, and further detected and identified by using the aforementioned high-resolution mass spec. Here is the ASMS data showing 13 off-DNA resynthesized compounds were assayed against SMN2, and risdiplam was pooled together as a control. Our data demonstrate that 8 out of 13 compounds bind to SMN2, where compound 10, highlighted in red color, shows the highest retention. We repeated compound 10 ASMS at different compound concentrations. The binding was reconfirmed in a dose-dependent manner, and from this plot, Kd can be estimated to be at 2-digit micromolar. Then, we did another assay, the fluorescence indicator displacement assay, using published RNA dye. Other than the RNA fragment presented in the DAO selection, the truncate with only terminal stem loop 2 was also assayed in parallel, in order to figure out whether compound 10 binds to this terminal stem loop 2. And the dose response curve on the bottom demonstrates that the binding of compound 10 to both RNA fragments with a consistent IC50 at what we can see in ASMS. So, we demonstrate a direct binding between compound 10 and SMN2 mRNA. But is this binding functional? To answer this question, we employed cell models to test whether compound 10 can regulate SMN2 splicing by rescuing SMN2 exon 7. We first conducted SMN2 mini-gene expression experiment by doing mini-gene transient transfection in HEK293 cells. In 24 hours, these cells were treated with SMN2 compounds. The treatment was allowed for another 24 hours. Then cells were collected, RNA was extracted. RT-PCR data clearly showed a band size shift in the risdiplam lane as compared to the DMSO negative control, indicating a potential splicing regulation. We later isolated this band. DNA was sent for single sequencing and was confirmed to be exon 7. Thus, our assay is approved to be effective in testing the function of SMN2 compounds. From this data, our compound 10 is less effective than FDA-approved risdiplam, but we can still see about 50% exon 7 inclusion, indicating compound 10 binding induced splicing regulation. To confirm this finding, we later obtained a fibroblast cell line derived from an SMA patient. This cell line was treated with our compounds directly, and RT-PCR was performed to evaluate exon 7 splicing regulation. In this experiment, cells treated with compound 10 for 6 hours did not show clear exon 7 inclusion, but when cells were treated for 24 hours, an obvious band intensity shift was discovered, which is statistically significant as compared to DMSO control. Our DAO screening thus identified a small molecule that binds to SMN2 exon 7 and can regulate exon 7 splicing with the potential to treat SMA. However, we also care about whether this compound is specific to SMN2. We performed RNA-seq experiment to check genome-wide gene regulation. Risdiplam was used in parallel as a control. We wish not to see too many differences in terms of gene regulation as compared to this positive compound risdiplam. We first looked at SMN2 exons. By calculating PSI, the ratio of the number of reads that include a specific exon to the total number of reads covering that exon plus the number of reads that skip this exon, we can see a complete exon 7 rescue by risdiplam. Our compound 10 still rescued exon 7 about 50%, which is actually consistent with previous RT-PCR data. We then looked at other genes. From these plots, compound 10 resulted in much more regulated genes with a fold change equals to 2 as the cutoff. When we consider both the fold change and the meaningful copy count change and set up the filter, we can see that compound 10 changed more than 8,000 genes, while risdiplam changed more than 2,000 genes only. Thus, compound 10 seems to be less specific than risdiplam, indicating further optimization of this compound is needed to increase the efficacy and also the specificity. Okay, I want to wrap up this story with a summary. We believe ShapeMap is a valuable technology in RNA research. It can quickly characterize RNA folding and serve as a judgment for your RNA folding by comparing the in vitro to the in-cell data, particularly useful for less explored RNA structures. Our specialized zipper DAL has been approved to effectively block RNA-DNA interactions, which enables RNA screening using this technology, which can cost-effectively and quickly generate a lot of binding information. Our SMN2 DAL screen showcase successfully identified small molecules that can bind to SMN2 functional motif and further regulate SMN2 exon 7 splicing. We can establish a diverse range of assays to study small molecule and RNA interactions. Our ALICE ASMS, coupled with the 300,000 HTS small molecule library collections, can be another powerful tool for you to identify RNA-binding small molecules. Our biophysical assay platform is well equipped with almost every biophysical instrument we can find. SPR has been frequently applied in client projects to screen for RNA-binding compounds, and in this case study, a client mRNA was screened against the 3,000 fragment library by using SPR. The hit rate is about 3.6%, and those fragments identified can be further validated in a separate SPR experiment. Well, the last take-home message I want to bring here is that we are expanding our toolbox to better support RNA drug discovery, relying on the knowledge accumulated from previous RNA screening using either DAL, fragment library, or HTS screening. We have collected building blocks, fragments, and compounds that are conductive to RNA. With this knowledge and the information from literature, we built a 4,000 compound RNA-focused HTS library. These drug-like compounds have been screened against 2 in-house RNA targets, resulting in hit rates of 3% and 12%. Several representative structural cores can be found here. We will be launching this service very soon. Our open access capabilities and technology platforms can deliver quality services to help customers achieve their goals more quickly, and we are now trusted by more than 6,000 customers. I would like to take this opportunity to express our sincere gratitude to all of our valued customers. We provided services, but we also learned a lot from our customers. And with that, I would like to stop here, and we are happy to take questions. Thank you very much for your attention.
Thank you, Dr. Zhifeng Yu and Dr. Jungyao Ma for that excellent presentation. And we do have some questions from the attendees. The first question is, what is the advantage of using DEL as an RNA targeting small molecule screening method compared to other methods, such as ASMS and phenotype screening?
Okay, so this is a very good question. So using DNA-encoded library to screening RNA targeting small molecules has its unique advantages. So for now, only a limited number of chemical matters that targeting RNA has been reported compared to protein targets. And DNA-encoded libraries has a huge amount of chemical diversities, and also it has a lot of structure novelties, and the screening was super high throughput. So it can give you a lot of information with a limited time, and also the DEL screening can generate extensive data sets reaching SAR informations, which is going to be super useful for the next stages and the medical care organizations. And also, if we compare the DEL to other screenings, such as phenotype-based screenings, phenotype-based screenings often yield compounds with indirect effect. Therefore, phenotype screening identified compounds always suffer from the target ID, and sometimes it's very difficult to find the true mechanisms. But DEL with the target-based screening approaches, so it helps you to find the hit directly binding to RNA target, which makes the downstream studies more easier. But still, we if possible still recommend our clients to use multiple screening methods together, since all the screening methods has its own pros and cons, so that if you combine different screening methods together, you can always increase the screening success rate, particularly if your RNA target is really novel. Thank you.
The next question is, RNA can potentially hybridize with DNA barcode. What is your strategy to eliminate the false positives in DEL selection?
Yeah, so this is also a very good question. Actually, our team spent a lot of time working on this DNA RNA hybridization issues. So actually, we came up with two different solutions. The first one was experimental treatment. It's just like Dr. Yu said in the webinars, we developed a specialized DNA-encoded library called ZipperDel to avoid the DNA-RNA interaction. But also, that's not enough. We also developed a specialized bioinformatics pipelines to analyze any potential DNA-RNA binding motifs coming up in the selections, so that we can remove them from the downstream compound selections. So by using both experimental treatment and bioinformatics treatment, we can avoid any false positive signals coming from the selections. Thank you.
The next question is about your RNA screening and folding validation. Do you have the capability of incorporating post-transcriptional modifications to the RNA target, such as methylated, acylated RNA? Many of RNA species have one or several of these modifications, and it tremendously affects its folding. And the attendee is wondering if you've seen a difference in the small molecules that were pulled down with naked RNA versus post-transcriptionally modified RNA.
Okay, thank you. So this is actually a very, very interesting question to me. So of course, for the first part, we do have the capabilities of doing those modifications to the RNA target, such as methylation, all those kind of modifications. But well, so far we only did some pioneer screenings against naked RNA targets. But our next step was trying to see whether the compound do have selectivities between the naked RNA versus the transcriptional modified RNA. And that's also a very interesting things for us to figure out next step. Thank you.
The next question is, how can we determine whether DEL-selected compounds are specific RNA binders, and how to avoid frequent nucleic acid hitters?
So this is so actually thanks for the questions. So for the first part, how can we make sure the adult like compound are specific RNA binders? So people always want all those hits are specific, not just non-specific binders, right? So to achieve this, we can easily set up parallel selection conditions in adult selection campaigns. For example, we can screening the primary target as well as its counter targets in the same down screening so that you can increase the possibilities to identify compound that is specific to a primary RNA. So we think non-structured mutant version of RNA are always the perfect counter targets based on our previous experience. Also, after you find any kind of potential chemotype and enrichment information after the down screenings, we always recommend you to resynthesize the compound and do multiple biophysics binding assays, such as SPR and MSTs. For example, in the SPR, if you see a very saturation curve, then probably your compound binding was specific. And also, using different methods can avoid assay artifacts. So we always recommend our clients to try as much binding assays together as possible. So for the second question is how to avoid the frequent nucleic acid hitters. So basically, we're trying to utilize the post-selection compound structure analysis from our chemical teams. So our chemical team would compare the potential delta-like compounds to our historical RNA selection data and our previous experience, so that we can see whether these compounds might be frequent hitters. So we are CROs, so we have accumulated a lot of experience in different projects and in also different screening methods, so that we can, by utilizing these pre-accumulated experience, we can decrease the possibilities to give clients a frequent nucleic acid hitter. Thank you.
The next question is about how you would identify false hits. For example, when separating binder and non-binder, if the DNA tag binds to the target, how do you identify it?
So basically, how we really identify a false hit. So, so I think this question is more about if the DNA tag was binding to the target, how can we know, right? So, like we said, so there's two ways the DNA tag can bind into the RNAs. So we using like I previously said, we have a bioinformatics pipelines. So and also we always include no compound control libraries in the selections. So we're gonna, after you finish your selections, we're always gonna go in and analyze the no compound control libraries first. And then by using the bioinformatics pipeline, we're gonna take a look to see whether there's any DNA sequence that is enriched in that control library. So supposedly the control libraries should be equally distributed in the DNA sequence. And if we see anything being an outlier, that indicates probably that's a false hit. And that probably coming from a DNA tag binding. And then we're gonna remove it from the downstream analysis for sure. So that's how we avoid the false hits. And also we do special treatment to the library for sure. So thank you.
The next question is, can ALIS, ASMS determine the KD value in a dose response experiment?
So our ASMS platform sometimes do the dose-dependent curve to determine the KDs. So you can use different dose of the compounds and goes into the ASMS to see whether it goes coming out of dose-dependent response signals. But sometimes the compound solubility is limited, so that you're not gonna get a very good KD binding curve. But in most cases, yes, you can do that. Thank you.
Can you make sure that the structure of RNA is right during the selection?
So just like Dr. Yu showed in the webinars, we have a lot of pre-selection QCs to make sure the RNA still maintains its abilities to bind its two compound. It's to make sure the RNA still folds into the right structures using shape map. And also we do a lot of like pre-selection QCs using SPR, MST, and all those kind of assays to make sure RNA structure was right. So I think yes. So after all those kind of assays was past QCs, you can kind of say, okay, my RNA structure is still there, and it retains its binding pocket.
The next question is, are there any specific reasons for choosing SMN2 pre-mRNA as a POC case study?
So yes, for sure. So, RNA structures are very, very different. A lot of RNA have very complicated and well-folded center structures. For example, those ribose switches coming from those bacteria, and already a lot of screening already have been done against these targets. But these RNAs, they have so complicated structures, but in reality, a lot of other RNA potential drug targets, for example, FOXP3 in tumor therapies, like Huntington mutant mRNAs, and also like SMN2 pre-messenger RNAs, they don't have that complicated binding pockets, but they are still identified as the potential drug targets. So actually, we choose the SMN2 as our POC target because we want to prove that the application of L is not just limited to complicated RNA, but also you can apply it to a lot of other messenger RNAs. And also another reason we choose for SMN2 is because it has been a lot of studies on SMN2. The downstream assay will be a validation to demonstrate that now it will be efficient, will be easier. And so that's kind of the reason why we choose SMN2 as our POC target.
All right, that's all the time that we have today. Thank you again, Dr. Zhifeng Yu and Dr. Junyao Ma, for your fascinating presentation. And thank you, participants, for being a great audience. Be sure to check CNEN or CNEN Online for information on the next edition of CNEN webinars. Thank you, On24, for technology and production services. And thank you to WuXi AppTec for the sponsorship that made this interactive webcast possible. For CNEN webinars, I'm Kelly McSweeney. Goodbye.